Yeah, happy to. So at Graphite Bio, we're a next generation gene editing company. We're using CRISPR technology. There are a lot of companies out there that have been making advances, using nuclease-based approaches, using CRISPR with the intention of designing therapies. We have a slightly different approach where what we're trying to do is use CRISPR essentially as more than a pair of scissors. We're trying to build on CRISPR technology to get to very efficient, reproducible, precise cutting and pasting, essentially, so that we can target a gene with a CRISPR nuclease, create a precise incision, and then instead of introducing errors, which is great for knocking genes out, in the case of knocking down disease-causing genes, if you have gain-of-function mutations, for example, and going beyond that and actually providing a template with instructions for how to really fix genes. So diseases like genetic diseases. Sickle cell disease is an example where we know the point mutation causing the disease. Beta thalassemia is another, where if we had the tools to do that, to do cut and paste, we could actually change DNA back to normal, restore normal genotypes, and potentially go beyond indirect approaches and offer really definitive cures to patients. And that's really our focus. We're doing this initially in hematopoietic stem cells, which we know can be one-time cures if they engraft in a patient. And our lead program that's in the clinic now is in sickle cell disease. So we had one poster on the clinical trial design for that. And then we have a new effort in our research that's in a research stage that's for beta thalassemia. And we also had a oral presentation on that program. Just briefly, the other areas that we're focused on is very complementary to the gene editing itself is better and safer ways of getting these therapies to engraft into patients. So we have efforts at the company where we're looking at targeted stem cell depleting antibodies that could allow these corrected cells to engraft in safer ways that's currently being used with chemotherapeutic type approaches. And we really are looking for having the biggest impact we possibly can with an exciting technology platform, thinking about patient impact. So our initial our lead program is in sickle cell. We have a research effort for beta thalassemia, and those are distinct because of the technology. Happy to go into why that is. Mm-hmm. And then we have earlier stage research efforts that are leveraging the same technology platform for essentially long-term, potentially permanent therapeutic protein and enzyme production. We're targeting alpha-1 antitrypsin deficiency for that early stage effort, and there could be other programs that would follow that. Yeah, that's exactly correct. So, I mean, in some ways, the sort of the first genome engineering experiment was in the 1980s, and Oliver Smithies, he's like delivered a piece of DNA and got recombination correction of an endogenous gene in the chromosome, and at that time he did this for beta globin. He did this really with the goal of correcting the sickle mutation, making that single change you're talking about. And the reason that that's so compelling, really, for the whole field is that if you think about genetic diseases where we know the cause, that are monogenic, that where there's also really inadequate treatment options and that affect a large number of people, 100,000 in the U.S., 60,000 in Europe, millions worldwide. This is probably the disease where gene editing can have the biggest impact. Sort of from a public health standpoint, and in terms of really addressing a significant unmet need. So I think that's the reason why there has been a lot of activity, a lot of interest in sickle cell disease. And the reason we're so excited about our approach and our platform is that we can go beyond some very interesting first generation approaches, which are indirect by nature, cutting and knocking out regulatory regions to try to upregulate fetal forms of hemoglobin. And we can actually go directly after the disease causing mutation now and try to rewrite that gene and correct it to normal. Yeah, so CRISPR is so powerful because it's I mentioned it's essentially a pair of scissors. It's a programmable modular nuclease. You put an RNA guide, direct it to the gene you want to target, and it creates an incision, and you can do that very precisely. Then the outcome that happens, though, is really depends on what the cell does once that incision has been made in the gene. And by default, what happens is the two ends of that gene are brought back together, through repeated cycles of cutting, re-annealing, eventually there are errors that are created. That's called error-prone repair, and that's the dominant repair pathway. That's what the initial wave of CRISPR therapies have been leveraging. Doing that intentionally, disrupting gene targets, knocking out genes. What we're doing is we're taking advantage of a different repair pathway in the cell, where the cell, if it sees a similar gene. Normally, this happens with a backup chromosome. When one chromosome is damaged, the cell can use a backup chromosome using a process called HDR, or homology-directed repair, and precisely, with extremely high fidelity, make a repair without introducing any errors. So basically, we figured out a way to use CRISPR, cause a break in a gene, activate this precision repair pathway, and kind of hijack it by delivering a synthetic template DNA instead of the cell using kind of a backup chromosome. It's using this template, and the template has the instructions for what we want to write into that gene target. In the case of sickle cell, that the key instruction is just that one single change that results in the normal glutamic acid being expressed in the protein instead of the disease-causing valine, just that one single misspelling. But our beta thal program we presented technically is even more challenging, because beta thalassemia is a disease that's not caused by one mutation. So it's not quite technically the low-hanging fruit that you mentioned. It's caused by hundreds of mutations. And our template in that case has to have an entire replacement gene that can be copied and pasted into the break we've made, and result in a normal genotype, a restored beta globin gene. So, a couple of points there. The first thing is, I think, to put this into context, these medicines are, if they can lead to definitive cures, are incredibly exciting. That we of course have to understand, and keep evolving our assays to really understand the quality of the cells that we're manufacturing, what off-target effects can be created. But I think in context, our cells are very good at handling DNA damage, and they're more double-stranded breaks on a transatlantic flight than with manufacturing CRISPR-based therapy. So I think it's important to keep that in mind when you're thinking about serious diseases. But that said, yes, we look at, we can characterize these products. There is increasingly a well-defined pathway with the FDA, especially for ex vivo products, where we can really do all the analytics we need to do and really understand the cells and their product attributes and the genomic safety before we infuse them into a patient. What we've thought about this in a few ways in terms of how can we improve on precision. So really two things with our platform that we focused on. One is using what we think is a better nuclease. So CRISPR-Cas9, of course, did not evolve to edit our cells. It evolved, as a defense mechanism in bacteria. So it's not perfectly specific. It's not as specific as it could be. So we're actually using an engineered version of that Cas9. It's called high fidelity Cas9. And actually we can measure that it has up to 100-fold lower off-target effects. So we think that's an advantage that can prevent things like cuts in two places and having chromosomal rearrangements or translocations. The other is because we're using this very precise repair process in the cell. So once we've repaired a break, that break is repaired and cannot be sort of a substrate for translocations or rearrangements, which can happen if you're getting repeated rounds of cutting and re-annealing. But I think big picture, to your point, there is a pretty clear path to kind of studying the off targets, characterizing them, understanding if there could be risk, and then making sure that we're measuring in patients, and collecting samples, collecting data, so that when potential safety events happen, we can understand them, put them in the context. So I think it really depends on the intended use. So for us, we're right now focusing, as we talked about, on where we think there can be big impact in the near term, and in diseases where we can correct bone marrow stem cells, get them to engraft. We're doing that ex vivo because we know that works. In that setting, we don't really care about the size of the nuclease. We want it to be as precise and specific as possible. So we would only really look at other nucleases if they're sort of better than this engineered version of Cas9 that we're using, and we haven't found anything like that yet. There could be other specific applications. like, for example, in vivo liver targeting with LNPs, where you really want to kind of shrink down the size of the nuclease to enable you to package it efficiently. Those smaller nucleases, smaller than Cas9, can have advantages from that standpoint, I think, in the in vivo setting. The other is the other, I think, very early stage exciting areas where we see that there can be CRISPR technologies that can kind of direct transposons or sort of other ways of potentially editing genes that are still in the kind of their infancy or prime editing, and it'll be exciting to see how those develop as well. Yeah, so I'm a cardiologist by training. I spent a lot of time in the lab as well, and after working on at Genentech on some cardiovascular programs and other programs, I joined a company called Global Blood Therapeutics (GBT) as the chief medical officer there, and led the development of it was a small molecule actually in sickle cell. So I really learned about the patient community, what patients experience, the degree of the unmet need. Worked with investigators and patient advocates on the development of a novel small molecule that was approved in sickle cell disease, and took a lot of those lessons. As I went to partner with Matt Porteous, our main scientific founder at Stanford, to build Graphite. And some of the perspectives that I bring are, as a physician and a drug developer, and very, very proud of the work we did to get an actual product approved. I mean, it's amazing to see prescriptions being written now for patients who really didn't have any new therapies for decades. I mean, really neglected area of drug development and investment, and patients who have really had inadequate options. So we had a real sense of mission and purpose, in specifically working to address a need that these patients have, mostly African Americans in the United States. And then now shifting gears to Graphite to actually be able to go potentially beyond incremental changes for these patients, and really be able to prevent what we learned were really the unmet need. So in sickle cell disease, a lot of people know if people know about the disease, they know that it can cause pain. Yep. And they know that it can send people to the emergency room and be very disruptive to their lives. But what's less well appreciated is that many patients with sickle cell disease don't even have these frequent pain episodes. And even if the pain episodes can be managed, there's really a devastating kind of relentless progression to this disease, where patients, die far too young. Life expectancies are still in the 40s to 50s, and there's really chronic, relentless organ damage that progresses. And the only treatment that can cure these patients is transplantation right now, bone marrow transplant, usually from a matched sibling donor. So the potential to apply a platform technology, really exciting molecular biology, but to a concrete medical clinical problem in a way that hasn't been done before, is what really motivated me to partner with our founders at Stanford and build Graphite around this technology and this specific unmet need for our lead program. Yeah, so the other data was on GPH 102, which is our beta thalassemia program. So sickle cell disease, beta thalassemia are two actually, more common monogenic diseases. They're both hemoglobinopathies. The genetic lesions are distinct, as we talked about a little bit earlier. And that's why we had to have, if our goal is go beyond indirect approaches and really change the DNA back to normal, correct the genotype, you need different approaches for these different diseases because the genetic lesions are very different. They both affect the beta globin gene, but in the case of sickle cell disease, every single patient has a single misspelling. So we basically designed this synthetic donor template to do this cut and paste and repair this single nucleotide and change it to normal, so that we could change a patient to be like a sibling with sickle trait, with the unaffected carrier status. So if we want to do the same thing with beta thalassemia, we actually need a different approach, because it's the same gene, but there can be deletions in the gene or hundreds of different point mutations in that gene. We can't make a different approach or different drug product, different template to correct every single one. So what we had to do in this case was design our template to have an entire replacement copy of the beta globin gene, to do this cut and paste and basically kind of pop in a new version of the beta globin gene to be able to normalize the gene regardless of the mutation or lesion in those patients. Technically, what was a challenge here is that if the template looks too much like the target gene, this repair process won't work. So it wasn't as simple as just putting the intact beta globin gene on a template to do this cut and paste. That doesn't work. We had to actually, think of a way of diverging the template and the replacement gene from the target gene enough, but still achieve the normal effect, a normal gene product in the end. And the way we did that was actually by diverging the introns. So we use introns from related gene, the gamma globin gene, which are close enough that they function totally normally, but they make the template look different enough from the target that we can get this cut and paste to work. Yeah, so we're seeing tremendous enthusiasm for the trial, and the reason for that is that there really are inadequate options in sickle cell disease, and there are a lot of patients who are severely affected. We're beginning with adults. If you think about bone marrow transplant alone, there are a lot of patients who are currently eligible and in need of cures by bone marrow transplant in sickle cell disease. Most of them, 90% of them, can't get that done because they don't have a matched sibling donor. So there are a lot of patients in need of this, interested in trials. Today we have more interest at our first 3 sites than we have spots in the trial. The ultimate goal, if you're thinking about making an impact in sickle cell disease, is to bring this to teenagers or kids, to actually cure a patient before they become severe. So that would be our ultimate goal. But initially we're looking at severe patients, and unfortunately there are a lot of severe patients who really are in need of these kinds of therapies. So there may be the opposite problem, which is when these products get approved, making autologous stem cell based therapies and scaling these up is not trivial. And it'll be a challenge for us and really for the industry to be able to scale up these therapies to address the number of patients who are going to be in need of these curative approaches.